Hello, good afternoon, good morning. So we are ready and we will start in 2 minutes. Hello, good afternoon, good morning. We will start in 2 minutes. Hello, hello. We will start very soon. We know that some people can be late, so we know very well the story. So we will start in 1 minute. So I think we can start. So hello, good afternoon and good morning for some people living in US. So I hope you are fine and ready for this interesting webinar. So I'm Didier Sampere, working in the Avantor organization in the global marketing team to push and promote chemicals and chromatography products. And I'm sure you will agree with me that the purity and homogeneity of AAV vectors are critical for the success of treatment delivery in gene therapy. And today we have the pleasure to present a webinar called Advancing Adeno-Associated Virus Characterization, High Resolution Techniques for Gene Therapy Application. And Ashley Kunz from Thermo Scientific will delve into cutting-edge methodologies for the precise characterization and quantification of AAV samples, focusing on the use of high-resolution size-exclusion chromatography and strong anion exchange chromatography. She will explore also the application of the new Thermo Scientific SurePac Bio 550 SEC MDi columns. Additionally to that, Ashley will cover also the use of the Thermo Scientific ProPac 32 SAX chromatography column for the separation of empty and full AV capsids. Ashley will bring you also many practical examples and case studies. And with that, I'm sure with all this information, I'm personally convinced that you will learn a lot on these new columns and techniques, and you will be more comfortable to separate such AAV substances. Before moving to the presentation, feel free to ask some questions, and using the box questions, we will organize the Q&A sessions after the presentation. This webinar will be recorded. I think that it's great for you because you will be able to share with your team and with your colleagues. And after, I will give you some details for the following after the webinar. So now I think that I would like to wish you a great webinar, and Ashley, up to you.
Awesome. Thank you so much. I'd like to thank the Avantor team for having me, and welcome to joining. So I am the Bio LC Columns Product Manager at Thermo Scientific. When you think about the portfolio that I work with, it is the DNA Pack, ProPack, MadPack, and then our new SurePac columns, and we'll be discussing those with you today. So to set the stage, we have, as many of you know, a ever-evolving biologics landscape that is getting increasingly complex, and this often happens because the molecular weight is increasing. And you can see here we have, monoclonal antibodies, which are a very common protein and very popular in the biologics landscape right now. But then we have other newer modalities such as AAVs, lentivirus, and adenovirus. And these are even larger and even more difficult to qualify and quantify in your manufacturing pipeline. And so when we are looking at AAVs, they are a gene therapy that are a viral vector that we want to insert some customized genetic coding inside the viral vectors and then be able to create personalized medicines and treatments for patients. And this can be very complex, and we want to make sure that as a vendor, we're partnering with you to ensure the safety and efficacy of these treatments as you go through the various analytical development R&D phases, through all of the clinical trials, and into commercialization and manufacturing QC. And so when you look at the genetic delivery of this material, you want to make sure that it's safe, obviously, but when it comes to the effectiveness, that's really going to be demonstrated by purity and the homogeneity of the AAV vectors. So aggregation is also going to play a significant role here as you think about transportation of these types of medicines. You want to make sure that they are not aggregating in a form that's going to be, by the time they get from one part of the world to another, that they are not going to be, bad for and causing any kind of immunotoxicity to patients, because aggregation is really not a good thing to inject inside of a patient. So we want to have that assurance and quality control. And size-exclusion chromatography is a great way to look at the aggregation of AAV stereotypes. And then we're also going to look into anion exchange chromatography that can be used alongside other orthogonal methods to characterize particles that are empty and full. And, this can be a replacement for other pretty expensive techniques such as AUC and that use a lot of samples. And often chromatography labs already have the HPLC equipment. So if all you need to do is change your column, then we can help you there from a separation perspective and gathering this quality data. As a reminder, AAV sizes is important when we look at these techniques, especially size exclusion chromatography. And so we're looking at about 25 nanometers in size. So again, there's a lot of different critical quality attributes, or CQAs for short, and we want to make sure that we are completely studying all of these areas of the manufacturing process as you go to develop and scale up into production. Having a good understanding of each of these areas of separation is very important. And so we're going to dive into these two application areas in specific today: size exclusion chromatography and ion exchange. And with that, we have our new SEC column, so a new product family for Thermo Scientific as well, which is the SurePac Bio. 550 is that's the pore size that is designed around AAV analysis, and then SEC MDi. MDi is standing for monodisperse inert. That's going to be really important in terms of the particles and the platform technology that we're utilizing in these columns, and then it's going to allow for that assurance in the inertness of the column body with that hydrophilic inert hardware. And the 3-micron particle of these new columns is going to have exceptional resolution, as we will demonstrate in our case studies for various AAV stereotypes. This product was launched on July 15th, and to give you an idea of what that looks like. So I think this picture really demonstrates the innovation here, as the monodisperse particle is quite unique and a solution we're really excited to be able to offer to our customers. So for size exclusion chromatography, you're really looking at separating the molecules based on hydrodynamic volume in solution. And the largest is going to elute first, and then you're going to see as you decrease in molecular weight, the peaks will elute later down the line in the chromatography. So this we have good applications for aggregate analysis and titer analysis, which we've already demonstrated is a critical quality attribute for AAV manufacturing. And so if you have your monomer, any kind of manufacturing related stresses, such as heating, freezing, transportation, it's going to really impact that, drug dosage, any kind of solubility there, as well as activity loss and undesired effects for patients. And so we want to eliminate this and understand these best case conditions for your therapeutic drug throughout every stage of the product life cycle. And so when you go into characterization, you're going to monitor the aggregation and ensure that you have a consistent product for safety and efficacy. So here's some attributions that we have for our new product line. So this is monodisperse 3-micron silica. As you can see in the additional SEM images, and a very common manufacturing in the past for chromatography media is this Gaussian distribution of particle size, and then sizing to the particle size you want to get to. So now with our new manufacturing process, we have a precise control over that silica and the size of the particles themselves, eliminating any kind of like fines in the process and other things that can be detrimental to the performance from a column to column and lot to lot manufacturing perspective. We have a diol modification on the surface of that silica because unlike other separation modes, you do not want any interaction with the surface of the silica and your analyte, because we want to only separate these analytes based on their size and solution and not by any other retention mechanisms that you see, like a reverse phase C18. And so the not only is the column hardware hydrophilic and inert, but we also have a modification on the silica to get that high purity silica and inert as well. And for the pore size, we have 550 angstroms. As we've demonstrated, this is the best pore size for AAVs and their size. And then we have 3 different column diameters, a 2.1, which is going to be great for any kind of SEC MS applications. We're starting to see that become more popular as new modalities are coming to the market, as well as a 4.6 and a 7.8. We went with 150 millimeter length here for multiple reasons, but when we worked with many customers, the benefit of the 300 millimeter length, which is double that size, is not really reflective in the return on investment that you're going to get with solvent usage and solvent waste. We want to be good environmental stewards and have greener methods. And so this is the 3-micron particle is going to allow you to still capture high resolution in your separation, but the 150 is going to give you a lot of other benefits downstream for cost of per injection of your analysis is going to be much lower, and we're going to have less waste. And so just thinking about these things in your method development as you move your method transfer from R&D and all the way through the different phases into manufacturing, this will really help you save time and money in your method and allow us to to keep that cost lower for the development of these materials. We also have guard columns that are offered, and then just keeping in mind some additional operation parameters. This is silica, and so your pH range is standard at 2 to 8. So if you go higher than that, you're going to start seeing some deterioration in that silica. You're not going to get as good lifetime out of your column media. And then for target molecular range and target species, certainly for AAVs, we really worked to design this around that particular size, but it can also work well for large biomolecules. So looking at that target molecular range based on what your analytes are within your developmental pipeline, looking at anywhere between 300 to 8,000 kilodaltons. So we also have seen some good data on, a 500 kilodalton protein recumbent vaccine. So don't just think, when you think SurePac Bio SEC, not just for AAVs, it will work great for AAV stereotypes, but it can also work for other large biomolecules as well. So for our recommended operating conditions, we really have found that the capillary impact is significant on your separation. So when you're going to develop your SEC methods, make sure you're considering the system volume and the tubing ID that you're utilizing within your separation, and also looking at a fully biocompatible UHPLC system. And so what does that look like to configure your system for optimum results? So you can see here we've used a 2.1 by 150 column, and there's a significant impact on the influence of the inlet and outlet capillary. So you can see our peaks here we go to some small molecule to just check the performance of the column. And when we take a smaller, more optimized capillary and fit it onto that system, we get enhanced resolution, as you can see here by the monomer and aggregate separation is going to be better. So overall, just keep in mind that the 3 micron monodisperse particle is going to give you more precise control. It gives us more precise control during media synthesis and column packing, but in turn, that's going to pass on significant benefits to you from column to column and lot to lot reproducibility throughout the life cycle of your development product, bringing products to market, and then commercialization. Again, that hydrophilic surface is eliminating any exchange sites on the surface of the silica, and with the 550 angstrom pore size being good for AAVs, as well as the hydrophilic coated inert stainless steel hardware. So for pore size selection, there was a lot of work done at Thermo Scientific to determine what was the best pore size that we were going to bring for our new product to market. You can see in the 300 angstrom, which is really better for monoclonal antibodies, other smaller proteins. The pore size is too small because you're missing those higher molecular weight aggregates. And then 450 and 550 look to be promising candidates, and so we dove deeper into those throughout the product development phases. And then we have a 1000 angstrom that's pore size is too large. You're not seeing good baseline separation. So really drilling into that 450, 550, and why did we choose 550? So we did have sharper peaks and better resolution with the 500 angstrom or 550 angstrom monodisperse particles. You can see a difference here in the chromatography from the top and the bottom images that even the smaller particle size of the 2.5, you get better resolution with a larger 3 micron particle size, but the larger, wider pore size as well. And so that's really where we felt like we could bring a better solution to market and drill down into that 550 angstrom. And so when thinking about detection modes and what to use, really we recommend utilizing multiple detection modes through SEC. So UV and fluorescence detection can give you different insights. So UV is going to help identify your peak identity, but then fluorescence is going to have higher sensitivity and will consume less sample, but if you don't utilize the UV, then you're going to miss out on maybe some of that extracellular DNA, additional impurities. As you can see, the difference between the two chromatograms here, the UV is providing some more insights into that. And then here we demonstrate some additional examples of various AAV stereotypes. So looking at AAV 3, 5, 8, and 9, where we were able to achieve baseline resolution of all the aggregates and monomer peaks for these different stereotypes. So flow rate choice is something to really think about in terms of method development and considerations for a parameter that you can control. So depending on what your needs are, if you want higher resolution, you may want to make your flow rate, decrease it down and get better separation in your chromatography. but then say you want to have a higher throughput method and consume less solvent and resources, you can increase that flow rate and still get great baseline separation. And so when you port that method into QC, it may be better to have higher throughput, higher injections in a day than that maybe, but in R&D, you're going to want some higher sensitivity, more details to understand the different manufacturing options that you have. So just think about flow rate as a choice of flexibility in your method development and determining what your end results are for chromatography and impacting that based on your desired needs. And so here's another chart just to kind of show you that impact of flow rate on runtime and resolution. And so you really have a lot of flexibility here to determine what you want to sacrifice and what you want to gain in chromatography. We all know that there's a bit of trade-off here and there depending on what outcomes you want to have. And so again, just this demonstrates that separation and how it can be impacted by flow rate across multiple different values. And here we have sample loading effects and injection volume. So we do have a large sample injection capacity with the SEC columns, but I would definitely challenge you and determine these samples are very precious. And if you can get a great resolution on a 1 microliter injection, then why would you use 40, right? So think about that when it comes to the types of samples that you have, and if you're sample limited, you can really get great quality data still with even a smaller injection volume in your SEC method. So SEC mols is also extremely important when it comes to AAV characterization. While Thermo Scientific doesn't have a mols within our instrument portfolio, there are other great vendors out there that do have that capability. So think about this when it comes to being able to get absolute molecular weight when there aren't always readily available AAV standards, or they're not extremely reliable. Mols can be utilized in terms of being able to get that absolute molecular weight on your AAV separations. So you can see here that we worked really hard to make sure that there was no shedding of the silica particles or any issues in terms of the detection, because for multi-angle light scattering, either columns that are out in the market, when you put them on the system, and if you don't run them for a very long time and clean off any of the impurities within that packing bed, you can see the detector just like lights up and has a lot of trouble with getting accurate, good data quality. So we worked extremely hard through our manufacturing process to ensure that out of the box, the column will work well on your mols detector and methods. You can see here that we've gone from a 4.6 by 300 column from our competitor, and we're able to transfer the method to the SurePac Bio SEC, and we have better resolution, and you can even see like that trimer peak is starting to come out a bit better because of that 3 micron particle size. So lots of benefits here, and definitely is good that this column will work with mols detection. So, as a summary, SurePac Bio SEC mols, SEC MDi columns are going to offer you high resolution and high throughput with that 3 micron monodisperse particle size. You have a wide pore size that's best suited for AAV analysis and other larger biomolecules of similar size. And then you have MDi technology for superior reliability. And MDi is going to allow for that monodisperse particle for the highest efficiency and best column-to-column and lot-to-lot reproducibility, and inert column hardware to eliminate any unwanted interactions and analyte adsorption. For more information, we have our part numbers listed here for the various regions globally, as well as we have a flyer and an application note. All of this content can be downloaded within the webinar resources, so please make sure to check those out and let us know if you have any questions. So moving on to focus in on ion exchange chromatography. We have our ProPack 3R, SCX, and SAX columns. So this is building on Thermo Scientific's 25 plus years of ion exchange chromatography technology. So really taking all the way back from 1998, we've had various ion exchange columns brought to market, and utilizing feedback from customers in terms of any pain points and issues. We really wanted to focus in on the development of these ion exchange columns to give the most reliable reproducibility, robustness, and resolution. And again, a lot of that is hinging on that monodisperse particle innovation. And so here you can see, again, a good representation between monodisperse and polydisperse. And so that historical way of manufacturing this media in that Gaussian distribution is really being completely changed with the monodisperse particle platform. And it's going to, again, allow for that improved lot-to-lot consistency. You can see it in the red circles in the right SEM image. You have a lot of fines and things that are inherent in that legacy manufacturing process that can create issues down the line. And so monodisperse is going to allow for a better packed bed stability, and often mode of failure for columns can be higher back pressure as you work, any packed bed instability and any of these fines that maybe didn't get cleaned out during the manufacturing process can then travel all the way towards the inlet of your column and then cause higher back pressure, and then you've got to get a new column and put more investment into that. So the monodispersed particles are really offering not only benefits to us from a manufacturing and increased capacity standpoint, but also to you as a customer, being able to have more reliability in the product that we can give you. And so we take that particle, which is a non-porous polymeric bead, and then we're going to graft that ion exchanger onto the surface through a cross-linked hydrophilic layer. And so taking all of that chemistry media and then packing it in a PEEK hardware, which is biocompatible by nature, not having any of the metals components within that column body. And so for more detail on the chemistry and the MDI particle, you're going to take that. We have the two different chemistries, the SCX, which is best for MAB characterization, and the SAX, which is going to be better suited for acidic therapeutic proteins, AAV characterizations, and any other columns that are going to have a PI or a protein isoelectric point of less than 7. And again, just honing in on that reproducibility, resolution, and robustness of the 3R. You have great lot-to-lot reproducibility is demonstrated here with infliximab, and high resolution that can utilize this SCX column with LCMS and get really great resolution of your adalimumab. And then for robust operating range, robustness can be thought about in a couple of different ways. Robustness in terms of column lifetime, which we can demonstrate in over 500 injections with our column, but also in terms of that flow rate and operating range as well. So whether you want high resolution, longer 45-minute method, or if you want to get that down to a rapid QC 15-minute method, the flexibility is yours, depending on that flow rate again. And so when it comes to how does this fit within our current portfolio, we will not be discontinuing any of our legacy ion exchange products, but you can see here that the ProPack 3R is the most advanced technology for ion exchange. And any new methods that you are going to be developing, please make sure to look at the 3R to take advantage of all of this new technology in terms of the monodisperse particle platform. For, again, that protein isoelectric point, if it's sitting at less than 7, you'd want to look at evaluating the SAX, so the anion exchange chemistry. And if your PI is greater than 7, you're going to want to look into the strong cation exchange chemistry. And so here's some data just to demonstrate across SCX and SAX, the various lots that we can show you good lot-to-lot reproducibility, as this historically can be a pain point for customers with other columns out on the market. And we really work to design in with the monodisperse particle platform, enhancing this lot-to-lot reproducibility and making sure that your charge variant chromatography profiles are as consistent as possible. And then again, going back to that robustness. So here, robustness in column lifetime. We have a good column longevity up to 500 injections for both chemistries. This is certainly modality dependent, right? And if you kind of, as the expert of your, your modalities within your pipeline, whether you have very clean sample matrix or not. So this can certainly be impacted on your specific target molecules, but just to know that we've designed in that robustness too, in terms of making sure that we can maximize the injections, and that cost per injection can be as low as possible utilizing our newest 3R ion exchange chemistries. So moving into some application examples. So we have here our SAX salt and pH gradients. So, certainly looking at that, the retention mechanism between SAX and SCX is going to be a bit different. You're going to see your basic peaks eluting first with the anion exchange. And, you can use a salt gradient, but also a pH gradient as well, which is going to help you predict that elution profile with a PI value. And you can have lower salt concentration in those collected fractions, depending on what you need to utilize them for. And so for our, we have various application notes that you can utilize that suggest starting methods, conditions, and optimized method conditions for your applications, depending on what types of modalities you're running. This one will dive into our empty full AAVs in particular. So again, looking back at that, that size of your AAV being around 25 nanometers. If you have a single linear strand DNA, you're going to have some additional weight associated there. So you can, utilize this SAX and anion exchange to determine your empty versus full capsid content. And here's what that looks like from a chromatography standpoint. So we have various AAV stereotypes that are demonstrated in the different colored chromatograms, so 1, 6, and 8. And your empty capsid is going to elute first. And then you have your full capsid as the largest peak, which is the most ideal, as you want to have the highest purity of your product. And so the higher that peak, the higher the concentration is. And then you can also see some impurities for some of the stereotypes versus others down the line in the chromatography. And you have your baseline separation achieved across all 3 stereotypes. And so for lot-to-lot reproducibility, this is utilizing an AAV6 sample, and you can see here that it's great reproducibility across multiple different lots of our column product. And so here is what we recommend in terms of a linear gradient with an isocratic hold. You can get even further baseline separation of the empty and full capsids. So make sure that you're considering your gradient profile when you're doing your method development. If you want to achieve better resolution, this is a parameter where you're going to be able to achieve that based on the gradient hold, and your system volume and total can certainly be impacted as well. So for our Thermo Scientific 3R columns, we have various part numbers here. We do offer in a 2 millimeter ID and a 4 millimeter ID with the lengths of 50 and 100 for both chemistries. Also keep in mind that the SCX and CX1 buffers are a really great combination to utilize. The CX1 buffers are a pH gradient, and it really simplifies that method development with a 10x dilution. And it's an area where we get a lot of feedback with maybe new technicians in the lab, or somebody that's making up buffers for a new time for the first time. That can be very impactful on your method. And if you don't have the proper pH, even just a 0.1 points of a difference can impact that separation and create some challenges with technical reproducibility. So the CX1 buffers are a good way to provide a little bit additional assurance and speed up your method development, as this is a linear pH gradient offering. Also, make sure to check out our additional resources for the ProPak 3R ion exchange columns. This will provide you the part numbers for the CX1 buffers as well. So additionally, for sample handling, we have a large portfolio with from Thermo Scientific outside of just LC columns. So for our large molecules, we're going to want to look into the SureStart polypropylene vials and closures. So we have, that again, going back to that analyte absorption, you want to eliminate that, because for glass vials, that can cause issues with sample recovery. And so with the polypropylene, that challenge is eliminated. And we also have our SureStart Web Seal Plate Plus glass coated well plates. So this is good with PTFE silicone mats, and we also have additional white papers and technical notes for our SureStart catalog of sample handling solutions. So for a review, we didn't get to discuss other new columns that we have brought to the market from Thermo Scientific within the last 2 years. But we did dive into the ion exchange with the ProPak 3R and the size exclusion with the new SurePac Bio SEC. But do keep in mind that we have other chemistries as well. So if you're working through peptide mapping, earlier this year we launched the Hypersil Gold peptide column, which has a media-specific QC with a peptide to allow for better lot-to-lot reproducibility down to that media level. And then when it comes to purification of oligonucleotides and even just analytical separation of oligonucleotides, we have our DNA-Pack RP chemistry, which is very unique and differentiated out into the marketplace. There's nothing like it. It is a super macroporous particle that is a polymer resin as well. So it's very robust in terms of pH stability and temperature stability. But then now we offer it in the larger semi-prep column format, so a 21.2 millimeter IB. So when you move that from analytical to characterization, you're able to now scale seamlessly between the different column formats from analytical to semi-prep, and fractionate your nucleic acids and do further characterization utilizing this larger semi-prep column. So for some key takeaways here, remember that these SurePac SEC columns are going to provide you high resolution separation of AAV and high molecular weight species for other large biomolecules as well. And then it can be integrated with UV fluorescence and moles detection for further AAV characterization. And then the ProPak 3R columns are going to be, help you with empty full AAV capsid separation with the SAX and utilizing those SureStart polypropylene vials for your sample handling options. For resources, you can go to the website listed below, select on your countries, click on the life science workflow, and check out key literature. And then additional key literature is included here within the webinar resources as well. So that is all that I have for you today. I greatly appreciate your time and attendance and would be happy to take any questions.
Thank you very much, Ashley. It was really, really complete, and I've learned a lot. So thank you very much for that. And I said, Ashley, we will open the Q&A session. So I've already checked and I've seen some questions. The first one from Italy is, what is the advantage of monodisperse particles?
Yeah, so really a lot of advantages, not only for us from a manufacturing standpoint and being able to provide consistent media, but from a chromatography standpoint, getting that maximum efficiency and lot-to-lot reproducibility, and it enhances more of that assurance over the lifetime of your method. So when you think about how long that process is for customers when they start in analytical development and R&D, and they go all the way through the different phases of clinical trials and into commercialization and manufacturing, that process can take 7 to 10 years or longer, and you want that assurance from year 1 to year 10 to year 30 through your commercialization and QC methods to be able to provide that lot-to-lot reproducibility and for that column that you get from us every time. And this is monodisperse particle platform is going to allow us to ensure that quality and reduce that variability from for the lifetime of your product life cycle.
Okay, another one. Can I use the SEC column for antibodies or other proteins?
Yeah, so it's really going to depend on the target molecular weight range of your modality. So for monoclonal antibodies, you're typically looking at 150,000 kilodaltons. And so 150 kilodaltons. And so when it comes to the size there, you're going to want to look at a SEC column that is a bit smaller than 550 angstroms. So really the MABs are best suited for a 300 angstrom size column. So we do have the MAB pack SEC1, which is a 300 angstrom. But if you're looking at something even larger, I mean, like, again, AAVs are 25 nanometers in size, but you have other larger biomodalities that are coming out within people's development pipelines that are in that 500 kilodalton or higher range. And this wide pore SEC column would be best suited for those larger biomolecules for sure.
Okay, thank you. Another one. What does inertness mean?
Yeah, so inertness in liquid chromatography is going to refer to the lack of adsorption of the analyte onto wetted surfaces. So the column really constitutes as the largest surface area within the instrument flow path. And so that inertness is important for several reasons. For biomolecules, many of the biomolecules are known to be sensitive to metals, so such as nucleic acids, oligos, and some other biomolecules. So that can pose a challenge from a sample recovery standpoint and just that assurance in terms of missing analytes. I mean, if you've ever seen a chromatogram that comes out and you're like, I was expecting a peak and I didn't see one, it's likely that there was some sort of corrosion or metal exposure that absorbed that analyte and it impacts sample recovery. So inertness can be beneficial for several reasons, but a lot of that is going to be around corrosion resistance and any kind of metal sensitivity, and that can be eliminated with the new hydrophilic coating on the surface of these stainless steel columns.
Okay, thank you. And another one, I think it's the last one. I hope that it will be clear. Can SEC column separate empty vs full AAV?
Yeah, so we would not recommend a size exclusion chromatography column for the empty full. This separation mode is going to separate more your aggregates and your higher molecular weight species based on size. So this is really why we pair the size exclusion chromatography column for aggregates, monomers, and titer with the AAV SAX column for the ProPak 3R. So the 3R ion exchange column is going to give you that proper separation mode to then be able to separate your empty versus full, which will not be able to be achieved by size exclusion chromatography.
Thank you very much, Ashley. So it was the last question. So thank you again for your great presentation. I would like to tell a big thank you to Jennifer, to Tiffany, to Florence for the organization of this webinar. And I would like to give you some details on that. So brochures. You have seen during the presentation some brochures and application notes. And I would like to encourage you to download some brochures and application notes, because you can go in the materials part in the process and you can immediately download these brochures. Or you have other possibility. You can go in literature part in our website. You can write chromatography consumables, Thermo Scientific, and you will find all the brochures and application notes created by Thermo Scientific. Okay, and you, of course, you will find these brochures linked to this webinar. In some minutes, you will receive a survey, and this survey is very important for us. So feel free to give us your feedback on this webinar, because, or your suggestion, because it's still important for us to read that, because we can improve the future webinars, or we can take your suggestions. Okay, tomorrow you will receive the recording. So I think it's important also to share this recording with your colleagues or with your team, because I'm sure that you have discovered many great information. So you will have to receive this recording. And the last the information is that next week you will receive a follow-up mail with application note, brochures linked to this webinar, and also links to the product in our website. So I think that this follow-up mail will be also important for you, and we will tell you a big thank you for your participation. Okay, so I think that it was a great webinar. Thank you very much for your participation. I can tell you that next year you will see other very interesting webinar made by our partner supplier, Thermo Scientific. So now I would like to wish you a great day for the American people, and the end of the day for the European people. Thank you very much again. And Ashley, it was great. Thank you.
Thank you for
Bye-bye.
having me. Have a good day.